introducing with the evening ATGC news, and today we have a major breakthrough in biology. Protein synthesis has been discovered, a process in which cells build proteins. Amino acids are linearly arranged into proteins through the involvement of ribosomal RNA, transfer RNA, messenger RNA, and other various enzymes. The central dogma is an explanation of the flow of genetic information. It is in itself the basic idea of protein synthesis. The dogma goes as following: DNA to RNA, then proteins, and from that, traits. In fact, every protein helps maintain our body. Without proteins, we would not evolve, reproduce, maintain homeostasis, make cells grow or develop, metabolize food, utilize energy, respond to our environment, and make DNA. Before diving too deep into the subject, it is useful to know that genes are a distinct sequence of nucleotides forming part of a chromosome. Now to Harrison with the actual process.
Thank you, Peter. The process of transcription is the first in protein synthesis, and I gotta tell you, this is fascinating stuff. In this process, which occurs in the nucleus, mRNA is built from an enzyme called RNA polymerase. The RNA polymerase uses DNA from the nucleus to complete this process. The newly assembled mRNA then carries genetic information from the nucleus to the cytoplasm, where it stays until it is needed for the next process. Groundbreaking scientific news. I'm Harrison Sailing, ATGC News. Back to you, Peter.
Now to Henry with more on the topic.
Thank you, Peter. This is Henry Schlesinger of ATGC News. After transcription, the mRNA that is made goes through a process that is called translation. Translation is a process in which a protein is made using mRNA. Before I explain this, there are a couple things you need to know. tRNA, T being an acronym for transfer, finds matching amino acids for mRNA. These amino acids are made from certain things called codons. A codon is a set of 3 bases that form an amino acid. This is why there are some cells that make up our heart and some that make up our lungs, but they have the same genetic coding. These cells are called specialized cells, which are a type of cell that performs a specific function in multicellular organisms. These cells work in groups to form tissue, muscles, and organs. What happens is the strand of mRNA that is formed in transcription is taken through the cytoplasm to a ribosome that uses tRNA to form a chain of amino acids that makes a protein. These amino acids are all made with the help of specific codons telling the tRNA which amino acid to make. That was translation and specialized cell types with Henry Schlesinger. Back to you, Peter.
Proteins are large molecules and they often have complex shapes. Protein structure is analyzed systematically by considering 4 different levels. A protein's primary structure is a unique sequence of its amino acids, which are arranged in linear order. The secondary structure is a regular pattern of hydrogen bonds between amino acid residues. A protein's tertiary structure is a characteristic overall shape assumed by the polypeptide chains. The quaternary structure is a 3D arrangement of subunits in a protein that comprises more than one peptide chain. Thank you very much. This is, as you can see, ATGC News. Hope you have a good night.